Good afternoon, everyone. Our guest speaker today is Caio Canella. Caio is an assistant professor of soybean breeding at the University of Arkansas. He obtained his bachelor degree from the University of Sao Paulo in Brazil, and has spent time at the University of Minnesota and Purdue University. There is his undergraduate degree. He obtained his master and PhD from the University of Missouri, working with Dr. Benjamin Chen, soybean breeder there, and previously at the University of Arkansas. In January of this year, Caio started as assistant professor of soybean breeding at the University of Arkansas, following the steps from his mentor. And Caio is running a very ambitious research program focused on topics from disease and herbicide resistance to biotic stress, such as water stress and flooding. His goal is to release new lines that can be commercialized and used by producers, and also plays a critical role in training new generation of breeders. We've had really exciting discussions with Caio during his visit so far, and we're very excited to hear more about your product.
Thank you very much. Very glad to be here. Thank you, Monse, for the introduction. It's my first time in Kentucky. I've never been here before, and so far has been a really, really great time. I had a chance to visit with students, with some faculty, and everybody has been extremely nice. So I really appreciate the invitation and also the receptiveness of everybody. I was debating a little bit of what I wanted to cover in this presentation because, you know, I heard that we don't really have a soybean breeding program here. So I decided to kind of give an overview of what we do in terms of breeding pipeline, what are our goals and how do we get to a final product, and then get a little bit more specific in some of the projects that we're working on. Hence the title, Challenges and Opportunities that we're seeing in soybean breeding when you look towards the mid-south of the United States, particularly in Arkansas. So going in overview of what we do, you know, our main goal is to supply Arkansas farmers with varieties that are tailored to whatever is their needs. And that can go from biotic and abiotic stressors, like Monse mentioned. And what we're trying to do is make a combination of a lot of the traditional breeding methods with our, you know, tried and proven, and they do work very well, with some combination of modern breeding approaches, which we'll try to cover a little bit more towards the data analytics, genomic prediction, genomic selection, and that kind of thing. So when we look into our breeding goals, I like to quote something that my advisor used to say, and Dr. Grover Shannon also used to say, that your first priority in a breeding program is yield, your second is yield, and you guess what, your third one is also yield. So at the end of the day, we can try to incorporate as many amino traits as possible, but the varieties, they do need to yield. So that's our main number one priority. We go through a breeding pipeline that is basically 100% conventional, and then anything that makes it to the finals, we go through the process of converting them to whatever herbicide tolerance platform farmers are interested on. So we have access to the enlist trait, we're getting access to the extend trait. So that's one way that we found that we could be much more competitive and have direct access to growers. One of the things that we really prioritize is this broad adaptation and yield stability. So we want to make sure that, you know, not specific to our target environment, but across several environments, our varieties are being stable and they're performing just like we tell the farmers they're performing. And one way we can achieve this particularly is on the biotic and abiotic stressors. So if you're breeding with those disease resistance, even though you get exposed to the stress, most likely you're going to be able to sustain your performance. So for us, the major biotic stress that we have to breed, and I imagine it should be somewhat similar to Western Kentucky, is root knot nematode, cyst nematode, and stem canker. Those are the three major diseases that we have to breed for. In terms of abiotic stressors, we go through all the water extreme stressors. So we're talking about drought and flood. We have active projects to develop soybeans that are adapted to either one of those conditions. We work with salt tolerance, so the chloride trait being either excluder or includer. And one of the things that I want to cover today is tolerance to off-target dicamba exposure. So that's part of what I did during my PhD, and we're continuing this work now in Arkansas. Besides this, there's also the value-added traits that we can work with soybean. That's why soybeans are quite an incredible crop to work with, because it's so flexible, right? You have high protein content, but you also have the high oil content. So there's a lot to play in between those two macronutrients. So we have traits for improved protein content. We manipulate a little bit the amino acid profile for specific targets, for poultry feed or aqua feed or those different industries, they require different amino acid profiles. We look at fatty acid profiles. So probably some of you have heard about the high oleic trait, low linolenic trait in soybean. We work on that. And something that we almost got into the discussion this afternoon, we also breed for food graded soybean. So natto type, edamame type, we look at the seed size, highland color, and that kind of thing for mostly for exportation, but also some domestic market. And this is another part that I had a chance to discover from Monse a little bit about this. We know that soybean genetic basis is extremely narrow. So we have a project to try to expand that by bringing diversity from, different geographical regions in the United States, but also from different countries. We know that genetic basis from the South is completely different from the North. So if you start breeding materials from those both places, you get pretty nice diversity. To give a little bit of numbers, I thought it was interesting for everybody. We went through some of those discussions today. In 2023, we released 2 new varieties. Those are maturity group 4. They're conventional. And before being released, those varieties go through extensive trials, across Arkansas in different states. We have our first round of enlist varieties, which I'm particularly excited about. Those hopefully are going to be hitting the market in 2025. And our advanced lines from our program that went through the conversion to enlist. So basically they have the same genetic background with the addition of the herbicide trait. And then in terms of numbers, so starting from the bottom here, we develop about 100 to 150 new populations every year. So those are biparental populations. All the crosses are done in Arkansas. We grow around 16,000 progeny rows. So those are 16 different genotypes that we grow every year. And then we input somewhere between a 5 to 10% selection pressure. So we'll move from 16,000 lines to about 900 lines that enter preliminary trials. In this stage, we put them to test for yield. So we are very fortunate to have a testing network in Arkansas. So for preliminary trials, they go about 4 locations. Those are all replicated trials. Depending on the performance, we move those to what we call advanced trials, which then they're grown in more locations. So this year we're using 7 locations, 6 in Arkansas, 1 in Missouri. And then from advanced trials, we move the very best ones that have the traits we're looking for and the yield potential to what we call regional pre-commercial trials. So here they enter something that the USDA has, which are regional trials, kind of like an unbiased assessment of variety performance, both from the public and private. And then if they perform in these regional trials, then we make the decision for release. So that kind of summarizes a little bit the process and the numbers that we do every single year. And as you can imagine, with breeding, it's a continuous process. So we start a new breeding cycle in 2023. That's going to be with us for probably another 5 years. But in 2024, a new breeding cycle will start it, and so on. So in 1 year, we may be dealing with 4 or 5 different cycles that started at different times in the year. Okay, so just to give you a little bit of images of what we're doing, this is our 2023 release. I hope the picture does justice, but it's a pretty good line, looks good in the field, well developed, and it has pretty competitive yield to the commercial checks of similar maturity groups. So there's a picture in Stuttgart, Arkansas, where we have our seed increase. So those are going through foundation seed. This is a picture of our enlist yield trial. So this is the first generation of converted enlist varieties. I'd like to think they're looking pretty good, and depending on performance this year, we're going to go through the process of increasing those and potentially becoming a commercial product by the end of 2024. So how is this done? Kind of put a little bit of perspective in our pipeline. The whole process starts with crossing, basic hybridization. This is where we're developing new populations. So like I mentioned, every year between 100 to 150 new populations are developed. If you remember from your genetics class, you make the cross, you get an F1. We need to go from an F1 all the way to an F4, so they're theoretically homozygous. This process to gain time, we do in winter nursery. So we use Puerto Rico for generation advancement. And now you start connecting some of the things we're discussing. If this process is done in Puerto Rico, it takes me about 18 months. If I was going to do this in the US alone, it was going to take me 4 years. So we are already shortening our pipeline, leveraging the use of off-season nurseries. And I'm going to touch base on this again, but what we're really trying to do is make the cycle as compact as possible, which will translate in higher genetic gain later. This come back as F5 about 2 years after a cross is made. Like I mentioned before, about 15 to 16,000 of those. We make our selections. They move to preliminary yield trials, 4 to 5 locations, make selections. They move to advanced yield trials, the 4th year. And then if they're performing well in this year, they go to those regional trials. And we end up getting a variety release between 6 to 7 years. It's a very typical, traditional breeding pipeline. Here is where things get interested, and that's where we're trying to, move our program towards, which is the use of molecular marker to make breeding decisions more efficient. So we know that the genetic gain for a given trait, you have your selection intensity, your accuracy, and your genetic variance. All of this is a function of time. So if I take 15 years to develop a variety, even if it's the highest yielding one, my genetic gain is really small. So everything that we're trying to do is reduce the breeding cycle as much as possible so we can maximize our genetic gain. And the use of genomic prediction is really critical because then you can start manipulating a lot of the variables in this simple equation just because of the availability of molecular markers. Is there anyone here that works with genomic prediction? Or if you could raise your hand. Is anyone that has no idea what it is? Also raise your hand. Okay, great. So what we do here is we use molecular marker information. Okay, just think about it's all the DNA of a plant. And you have a training model and a validation model. My training model consists of molecular markers and yield data. So I train my markers based on observed yield data. And then what we try to do is we generate molecular markers for lines that were not tested yet. And then I predict how they're going to perform based on molecular markers alone. And as you can imagine, if I can do that, the cost of my program goes down significantly because field trials is what really drives the cost. Phenotyping is much more expensive than genotyping nowadays. And so the cost thing is one that is really critical. But think about, again, if at the beginning of my cycle, I can genotype my materials. First of all, I'm going to know my genetics pretty well. It's not guessing anymore. We know exactly what we have there. And then I can go through this process of prediction, which will help me to skip some of the testing years. And then going back to this equation, the more you start reducing your cycle, the higher is your genetic gain. And this is something that the companies are doing, routinely. And that's what we're trying to implement in the program now. So really leveraging data to make more efficient decisions. So again, very overview of what it is. You have observed phenotype plus markers. Train a model to estimate the effect of the markers, and then you're going to have an observed phenotype that will be predicted based on the markers. That's the 30,000 view of what genomic prediction really is. So if you can do that, this is how a predictive soybean breeding pipeline would look like. We can go through crossing block. This is not going to change. That's the basics of breeding. And we're not trying to reinvent what breeding is. We're just trying to use technology to make the process more efficient. At the end of the day, the principles of breeding are very well established and they work. We go through crossing, we go through generation advancement, nothing changed there. The one thing that changed, we removed the progeny row stage. And as if you recall, progeny row stage is where we have the highest number of genotypes. And we do not have yield collection in that stage. So I'm replacing 1 year of trial that's basically visual selections. There's not necessarily data with genomic estimated yield. And then I can cut that stage and again, make my genetic gain higher. So that's one of the main things that we're trying to do. And if you recall, the previous process took between 6 and 7 years to get completed. With genomic prediction, we can start releasing varieties within 4 years. So again, your breeding cycle is much shorter, meaning your genetic gain is going to be much higher. And breeding, like I mentioned, is a continuous process. So the sooner I can identify superior varieties and start crossing with them, the higher the genetic gain my program is going to be, because this process of recycling genotypes is going to be done much faster and more efficient. So that's a very overview of how this can be beneficial. And someone may be thinking, okay, how well can you predict, right? What is your confidence to make your predictions? So I want to bring one study that we done last year, and I had a chance to discuss a little bit with Monse. And again, since we have a very diverse crowd, I'm not going to specify too much in terms of the genetics, but here we used almost 800 breeding lines. So those are all genetically diverse breeding lines. We test them over 5 years, so we have pretty solid yield data. And we use those varieties to train a prediction model. And then we estimated in our own varieties how well this prediction model is working. The difference with this one is, like what I describe as genomic prediction is very simple. At the end of the day, it's much more complicated than just using marker effect. What we try to do here is identify some sort of covariate that would be consistent across years. We could use the environment, but the environment, you cannot predict what's going to happen the next year. Or you can, but not super precise. So what we did here, we use soil texture, which is something that is somewhat consistent over the years. And then we use that covariate to help estimate better the effect of the markers. So meaning that, okay, if I'm going to be planting those varieties in a sandy soil, this is how they should be performing based on the soil texture. If we're going to plant in a loam soil, this is what's going to happen. And that opens opportunities to do a lot of different covariates. For instance, physiological covariates per environment, root growth rate, or something else that is specific to your environment. What is nice, and again, we could discuss this all day. I want to try to be quick here. We look at the phenotypic variance within and across environments, and how much adding the soil covariate help to distribute the variance. And you can kind of see when you just have a simple model with the effect of the markers, you have about 65% goes to the arrow term, the environment term. And you have about 25% is just residual that you cannot explain. So there's a lot of phenotypic variability that you're not accounting for. And then you see when you incorporate soil plus the interaction of markers with the soil, the amount of phenotypic variance attributed to the environment goes down significantly. You go from 65 to 45%. Your residual, your non-explained variance goes down a lot as well. So we start almost structuring this error term, meaning that the estimates for your markers are going to be much more precise. And then when we look within environments, we can kind of see something similar. The residual goes down tremendously compared to the very basic GBLUP model. And you can start structuring your terms much more precise than if you did not have it. So at the end of the day, with soil texture, it seems to work very well. And it opens opportunities to do with many other different traits. In terms of accuracy, we go through multiple cross-validation scenarios. And again, not to get too into it, we basically predict observed lines in observed environments, unobserved lines in unobserved environments, and vice versa. So there are several combinations we can do. As you can imagine, when lines were observed prior, your accuracy is usually the highest one. So we can predict about, with 0.6 accuracy, it's pretty decent. If I can't throw away most of my lines with this much accuracy and just focus on the ones that have high yield potential, we're making progress. And then when things start getting a little bit tougher, when you start trying to predict unobserved lines in tough environments, your accuracy goes down a little bit. But one point that I want to bring up here, and if someone is working with genomic prediction, is this concept of practical accuracy. We get too tied up with correlations when at the end of the day, for the breeding program, I want to identify what's the highest yielding, not necessarily the whole group. I want to see if my model is picking the best lines. And that does not always align with just correlation. So what we've done here, we're breaking down by yield potential. So we look at different environments, and then we classify lines as top 80%, from 60 to 80, from 40 to 60. So we created yield bands based on the test performance. And you can kind of see that when you use genomic prediction, you have very high accuracy classifying the top yielding ones, and vice versa, very high accuracy classifying the bottom ones. So for instance, for genotypes that are performing 80%, the top 80% of the whole trial, about 71% of the observed genotypes were within this group. So we have pretty nice accuracy. We're not really missing anything. When you look at the bottom one, most of the things that were predicted to be on the bottom yielding were actually the lowest yielding ones. So moving away a little bit from just correlation and looking at practical application, this seems to work extremely well. And we're really excited to be deploying those models in the actual breeding program. Looking at the most tough condition, unobserved environments and observed genotypes. We have no prior data for any of these. Obviously, your correlation goes down significantly because you've never seen those lines before. But then when you start looking at this distribution based on yield performance, the model can pretty well identify at least what is the half most yielding, the half lowest yielding. And again, for this stage of progeny rows, imagine if I can move away from testing 15,000 genotypes in the field and test, a handful of those based on genomic prediction. That's going to make my program much more efficient. We're going to have much better allocation of resources. And breeding is kind of like this balance between resources and what are your outputs. So the more efficient we can be, probably the more gains we can make. And last thing that we did, not convinced yet, we wanted to see based on the breeding decisions. So like I mentioned before, we have lines in preliminary, advanced, regional, and then uniform trials with the SDA, and then lines become released. So we color the similar plot here based on their fate. Were those lines released? Were they in the pre-commercial? Did they die in advanced, or did they die in the preliminary stage? And we can see here, probably the slide is not making the justice. When you use the model, you do not discard anything that was a release. So again, let me rephrase this. Coming from 15,000 genotypes, if I'm using genomic prediction, I may lose some high yielding lines, but I'm not losing anything that would become a product. And from the breeding standpoint, to me, that's 100% accuracy. My goal is to identify what's going to be released, not what's going to be sitting on my cold room. And the model seems to do that pretty well. So we have very good accuracy in identifying what is the highest yielding. There are multiple other things that you can do with genomic prediction, but I didn't want to make the talk just about genomic prediction. We're using it to change the way we do trials. So you think about, you have 3 replications, you may move to 2 replications and predict your third. You can do a subsample of genotypes in each environment, which is what we call sparse testing. There's multiple things that are being developed with the use of genomic prediction to make better breeding decisions. But when you put both of these side by side, you can kind of see, you're cutting here about 2 years of your breeding cycle. And probably cannot read, but if you go to the paper, will be easier to see. The number of data points that you're actually generating is much higher when you go through predictive breeding, because I'm now wasting my resources in an enormous number of genotypes. We're wasting our resources in genotypes that have any sort of potential. So we're directing our resources for things that have a chance of becoming a product. Okay, this is what I wanted to cover related to what we're doing in breeding, just specific breeding. I don't know if there's any question, or we can leave questions to the end. But I want to bring the work that we're currently doing with Dicamba tolerance. So I imagine probably not much here, but in Western Kentucky, at least, this must be a big problem for growers and researchers. We heard some stories that you're getting trouble with the application. And that's what we were observing back in Missouri. Too much off-target exposure, and we tried to develop a research out of it. And at the end of the day, ended up being pretty nice. So I just want to make it very clear that the technology per se is really nice. It's sound research. I believe it was published in Science back in 2006. At the time where you didn't have any option for weed control besides glyphosate. they come up with this as an opportunity to maintain biotechnology-based weed control. So there's a lot of good rationale behind it. The way it was developed was really interesting. They were observing that in the soil, dicamba was being very quickly deactivated, and then they investigated the bacteria in the soil. They identified what was converting active to inactive dicamba. They isolated that gene, put it in the plants, and surprisingly, the soybean plants were tolerating, 10 times the label rate. So it's a really nice work that was done in Nebraska initially. Monsanto obtained the technology, I believe, 15 years ago. And then in 2016 is when the first extant variety came to market. And as you can imagine, there's a limited amount of herbicide options. So in the first year, you reach almost, over 50% of the whole market. It really took over ever since year one. The problem is that, we call it the aftermath, because we weren't really expecting that dicamba would start volatilizing that much and starting to affect, off-target crops. And that's I mentioned to some, this is a poster from Corteva I saw at the SID meeting in Chicago that, they're emphasizing that the least trait, you're hitting your target, not your neighbor's field. So it's kind of interesting how they were marketing those two technologies. But anyway, between 2017 and 21, over 10,000 official reports made to the EPA, and the USDA estimates that to be, much higher than just 10,000. It really became a big problem in the Mid-South. And as another statistics from the USDA, that nearly 40% of fields grown with extent never really sprayed dicamba. So you get you thinking that maybe farmers are choosing this technology almost to protect themselves from possible off-target exposure, which, the numbers. So how this started, I remember walking plots with my advisor. We were right in the Boot Hill, Missouri, so a lot of exposure. And we're seeing a lot of damage, but we run into something like this, where this is a conventional variety that for some reason was showing way less symptomology than something like this, which was also conventional. And as you can imagine, this is the extent check. What's interesting, we started seeing this across multiple fields, and then we wanted to see if there could be genetics. We went to our progeny row stage, and if you remember, progeny, you're crossing one parent with another parent. And we started seeing segregation for the response to dicamba within a population, which for us was a big indication this could be a genetic trait. So very susceptible sister line, very tolerant sister line. And then we went to the germ plant collection and brought in a lot of genetically diverse soybean accessions. Those are all over the world, maturity groups from 3 to 5. And we started observing similar response. We have some accessions with, very minimal symptomology, and side by side with one that showed severe symptoms from dicamba. We were a breeding program, so simultaneously we were developing materials with this dicamba tolerance. And this is a commercial check that was a Roundup Ready at the time, and this is our conventional dicamba tolerant. So it's really nice to observe them side by side. It's almost like half was sprayed, the other half was not, but they're exposed to the same environment. So this is one of the advanced lines that we're moving forward as a dicamba tolerant genotype. And then, you identify something, you want to develop a mapping population to confirm if what you're observing is really true. So we crossed some of those diverse accessions with our susceptible breeding lines, adjusted breeding lines to the program. And, exciting enough, we start seeing the same level of segregation in the progenies developed from those two crosses. So another big indication for us that this was really a genetic trait. And here's how they were looking this year. You can kind of see one of them, free of symptoms. You don't see any sort of dicamba symptom. And the other ones, you start seeing all the typical, cupping and curling of the leaves. So one of the things that we were considering is, okay, we're seeing this visual difference. Does this translate in yield? Can you offer some sort of yield protection when you're growing a known extent variety under dicamba exposure? So we have a relatively large experiment, a little over 550 different genotypes. We've grown them across several environments. We got about 7,000 data points across all those years. So it's a pretty decent data set. We did a visual score just based on the appearance from 1 to 4, and all the plots had yield data. So again, pretty substantial data set. One of the questions that we always got related to this is, okay, how do you know your dicamba exposure is homogeneous? And it's really hard to do. We really can't control the exposure coming from so many sources. What we decided to do is use the same genotype from the companies, one being a run-up ready, the other one being an extend. So it's basically the same genetics, same maturity group. The only difference between them, one is run-up ready, the other one is extend. Assuming that if there was difference in performance between them, that's due to dicamba exposure. And then we had those, spread across our field. And then we were able to develop some sort of yield maps based on the difference between glyphosate and extend checks. And pretty nice, we could see that the dicamba distribution was significantly homogeneous across all those environments, meaning that you spray it in one location, it travels to another one, and it precipitates kind of like very uniformly across your field. So that one was one of the questions that we got that we were able to prove. The other one was, okay, how accurate is this visual score? We didn't have any necessarily protocol for phenotype. It was something we started. So we used two different metrics to confirm that what we were observing is really consistent. One of them is this Cronbach alpha, which looks at the consistency of your phenotype across multiple observations. You basically go from 0 to 1, the higher, the more consistent. You can see that we got from the Cronbach from 0.93 to 0.96. That means that if I was giving something a 3, most likely that was getting a 3 across multiple environments. Pretty consistent phenotype. And then we look at the correlation within and across environments, which, the darker the color, the higher correlation. You can kind of visually see that most of the observations are pretty correlated. Again, if you got a low score somewhere, you got a low score somewhere else, which again, for us, very good. If this was all over the place, a lot of what we're seeing is because of environment. If it's this consistent, a lot of what we're seeing is because of genetics. And then, the final question, does it affect yield? So what we did, we adjusted the yield of every one of those, 7,000 data points to the nearest extend check in the field. So we would minimize any potential spatial variation or anything. It's nearly, neighbor plots. So if it's performing here, you assume it's the same environment. We saw that as you increase the damage score, you're going to see, on average, about 90% of yield. That means that you may grow a conventional line with tolerance under dicamba exposure, and you may observe about 90% of yield loss. At the same time, if you're growing a highly susceptible conventional, you may observe almost 40% of yield loss. So it does provide a pretty nice yield protection when you're being grown in environments like this. The other thing we did is, we saw that there was a clear interaction between maturity group and this yield variation. And that's something that we heard, probably your later maturity, they have a longer time to recovery. You're going to see that they have less dicamba susceptibility, which at the end of the day would mean that all we're seeing is difference in maturity. So what we did is, and that's where we got very excited, we plotted the response based on maturity group. So let me walk you through this. We have dicamba scores here. If you remember, go from 1 to 4. And then each bar is maturity group. So you can see that within the tolerant group, they all perform the same across maturity groups. And as you go through the susceptible, then you start seeing this difference in performance. That means that within the tolerant group, the reason why they're sustaining performance has nothing to do with maturity. It has to do with the fact that there's some physiological mechanism regulating this response to dicamba. Whereas for the susceptible group that does not have this trait, the difference in yield is because some have longer time to recovery than others. All right, maybe tight on time. I'm gonna go faster on this. We used a drone to try to classify all those varieties. As you can imagine, walking 7,000 yield plots is not very fun. And if you can use a drone to do that phenotyping, it's much nicer. So we did a very simple experiment running just an RGB camera. There's nothing fancy there. And we used about 230 soybean genotypes. They're pretty diverse, different responses to dicamba. And we combine image features with two different machine learning models. So one of the artificial neural network, and the other one is a random forest. Those are the features that we extracted from each image. And what we try to do is kind of replicate what we're seeing with our eyes. So for instance, you look at the coverage of a tolerant line and another one from the susceptible. Probably the first thing that comes to your eyes is what? The canopy coverage is very different between them. So the susceptible one, you would imagine they have lower canopy for multiple reasons, from the cupping and anything else. You can look probably not very well from there, but the intensity of the green color is very different. When the leaves go through cupping, it changes the color of them. We look at the texture of the plot, what we're referring as entropy. You imagine when you get the damage, they get all rough, right? You have all the cupping happening. When they're tolerant, they're always smooth. So we try to replicate using image features the things we were seeing with our eyes. And it was kind of cool when you look at the distribution of those features within each class, they all make complete sense. So like I mentioned before, you look at the canopy coverage, the highest value significantly different is from the tolerant group, 0.6. The one in between is from the moderate, about 0.2. And the one for the susceptible group is the lowest canopy coverage. And again, these are all z-score adjusted values. We look at contrast. We look at entropy. We look at some vegetation index. We look at the saturation of the color. All of those kind of follow the same observations we're expecting. The tolerant showing some specific characteristics, the susceptible, some specific characteristics. All right, so we run, like I mentioned, two different models, one with the artificial neural network, the other one with random forest. Our overall accuracy, and again, keep in mind that this is very little control on the experiment. We're relying on off-target Dicamba exposure and visualized as ground data. For me, getting this accuracy was really nice. I think it's pretty nice, and for what we need to do, it's more than enough. But in terms of practical application, we wanted to see how well were we classifying the tolerant and the susceptible ones, the two extremes. And if we were making any misclassification, like it's a tolerant one, but we classify as susceptible. With the A and N, we didn't see much. There's, some misclassification, extreme misclassification, but not too much. When we moved to the random forest, they performed much, much better in terms of misclassifications. So you can see when you're predicting to be tolerant based on the image features, you make some moderate misclassifications, which are not too extreme. If they're moderate, that's okay. But on the susceptible side, we're not making any of those. So again, to me, model works extremely well for this high throughput screening of thousands of genotypes. Are they susceptible, moderate, or tolerant to Dicamba? And then, moving forward, we wanted to see if the genetics made sense. We had two separate populations. We have one based on breeding lines, which is a yield study, and we have the one based on the PIs, which is what we use to develop populations. So we did a GWAS study, so genome-wide association study, and we want to see if there's some regions of the soybean genome that are associated with this differential response to Dicamba. We have about 50,000 markers, so it's a typical genotyping platform that we use. We have 2 years of data, and then we use two different models. One was our benchmark with the B-Link, which is very used in GWAS studies, and we developed this G/E model. And how this works is this: we obtained a population structure for all of those genotypes. So you do this through PCAs, and if there was population structure affecting this response, and that was because of the environment, we did the interaction of each one of the principal components with the environment term. So that would give us a much better estimate of each one of the molecular markers when accounting for this variation. So that's kind of just an overview of how that was done. When we look at the results, I mean, if you're not in genetics, how this works is, and this is called a Manhattan plot because there's a bunch of peaks, hopefully. On your y-axis is the significance. The higher it is, the more significant it is. And your x-axis is the chromosome. Soybean has 20 chromosomes. So the high in each one of those dots is one molecular marker. Like I mentioned, we had 50,000. The higher they are, the more significant to the trait they are. So here's where it gets interesting. When we run those two models, we have overlapping associations that were identified in chromosome 19, so it would be this one here, and one in chromosome 10, which is this one here. And then when we look, we can go to database and see which genes are underlying those regions. We saw that the one in 19 is associated with glycosyltransferase genes, which, when you look at weed science side of things, this is associated with detoxification of plants to herbicides. So we were very excited to see that some of our hits end up being, making biological sense. The other one was a multi-drug resistant protein, which is also associated with detoxification. It's the last phase of detoxification, which is simply the translocation of this conjugated herbicide into the vacuole. So it, for us, was really great because what we're seeing is recovery of plants after being exposed to dicamba. Some recover much better, some doesn't. And based on the hits that we got, it makes a lot of sense that for some reason, some genotypes, they have a better ability to recover from the dicamba exposure. And here's just a description of what is phase 2 and phase 3 of detoxification from herbicides, which is exactly the genes that we found with those hits. And then when you look at the phenotypic distribution, we created some haplotypes combining the significant markers. So, you have 3 different SNPs, you have 6 possible haplotypes, and you can see very clearly, here you have all unfavorable alleles. So none of those are associated with tolerance. As you increase the number of favorable alleles, your score goes down very clearly. And the lower score means less dicamba damage. So we really think this is a quantitative trait, but you can explain a lot of the variation with just a handful of markers, which should be, a focus later on in some studies we're doing. This is the last thing that we did, is a paper we published just 2 weeks ago, I think it came out, which was understanding the genetics from a completely different breeding population and seeing if some of those results from the PIs make sense in the breeding population. And then we developed this GWAS model based on some machine learning algorithms. We're trying to do some different things here to see the lowest number of SNPs that are going to provide the higher accuracy. So don't think I have enough time to go too deep into this, but think about, we had 6,000 markers and we have 500 observations. If you remember from statistics, your number of samples is much lower than your number of predictors. You end up running into a problem of overfitting. You just have too many predictors for too little samples. So we want to do a variable reduction. We run a partial least square, we calculate the variable importance, and then we discarded any SNP that did not have any association with the trait. That was number one, featuring process in this analysis. And then the other problem you run in data science is when you have multicollinearity, meaning that you have highly correlated predictors. They're going to affect your model's performance. So we look at the correlation between each one of the selected SNPs. We discarded the ones that had, very high correlation, meaning that the SNPs that were used for our analysis were the ones that were very significant with the trait. They were unique and they're not correlated among themselves. So we have like a pretty clear set of predictors. And then how this thing works is we were fitting one SNP at a time, kind of like a stepward filtering process, and we were printing the classification accuracy. So you have one SNP plus another one, this is your accuracy. This plus a third one, this is your accuracy. You want to see what was the very minimal number of SNPs that would confer you the highest classification accuracy. And this is kind of what we get. So we had about 40 SNPs filtered from those initial 6,000. And with 12 SNPs only, we got almost 80% classification accuracy, which to me, that was really nice. I mean, you would imagine that the more SNPs that you have, the more accuracy you're going to have. But we show the data that's not really the case. The more predictors you end up having, you start almost modeling your error term. And the error is no longer reproducible. So you're really getting lower, classification accuracies. This was with the support vector machine model. And then we did with another random forest model, very similar. With 17 SNPs, this one we achieved the highest classification accuracy. Looking at, visually the precision of this model, you have on your x-axis the predicted class. So for instance, here's a tolerant, moderate, and susceptible. And then within our predicted as tolerant, 86% were observed as tolerant. And the remaining was observed as moderate. So we don't have any extreme classification. When you look at the susceptible class, 78 were predicted as susceptible and observed as susceptible, and about 20%, moderate. Again, no extreme misclassification. So for us, again, practical application, you can do a really accurate filtering of your lines based on genomic data, which is what we're looking for. And then this is just I'm sure some people think, okay, what if you fit all your SNPs? What will be your accuracy? And we saw a pretty nice trend. You get your highest accuracy, and as you increase your number of SNPs, your accuracy start going down. And we run something similar for root non-nematode, which is much more qualitative than this, and you have an even higher decrease in accuracy. Goes to show that it's very nice to have a lot of markers, but when you're actually going through your analysis, overfitting is a big problem. So, you know, you look at data science, feature engineering is almost an entire degree nowadays, and we really need to start doing some of this simply breeding to get the highest accuracies. That's what I have. Some of the summary and conclusions is deploying genomic prediction in a breeding program can make a big difference in terms of your resources allocation, efficiency of the program, and overall genetic gain. Models are important, but the one thing that really drives genomic prediction is the quality of your phenotype. Sorry. To make genomic prediction works well, it needs to be beneficial, meaning where are you applying this? What are you planning to get out of it? If you don't really know what you're trying to do, it costs a lot of money. So if you're not really capturing the benefits of this, it's not really worth it. You're going to spend, you know, hundreds of thousands of dollars to not really see much gain coming out of it. When you look at the Dicamba thing, we know that it's not just cosmetic. I remember when the technology came out, there was a lot of discussion whether it was just cosmetic, whether it could even give you a yield boost, or if you had a yield loss. From our data, which is an extensive data set, it looked pretty clear that you're going to see substantial yield loss when exposed to Dicamba. One of the main findings that I think is important is we identified that the genetic background does make a big difference. So you may have non-extend varieties that perform differently to Dicamba. And that opens up a lot of opportunities. One, you're growing conventional, you can coexist with this Dicamba system. The other one, you may be growing an enlist variety that can also coexist in this environment. So it gives you more flexibility in the choices of variety choice. UAV system, very simple, especially for traits you can clearly see. You don't need any fancy camera. You can get by with, you know, a lot of information from just an RGB camera, especially if the trait is very clearly distinguished by your eyes. And lastly, it's a long sentence. I'm not going to read all of it, but what we're trying to say is that genetic resources that are conferring tolerance to Dicamba, it not only helps to sustain a conventional system, but it gives you a lot of flexibility in different systems. So especially for us in the Mid-South, this is very important because we get a lot of target exposure until this date. Okay, that's what I had. Thanks again for the invitation, and very glad to be here. If there's any questions or comments, I'm happy to hear it. Thank you. Yeah.
I learned a lot. Thank you. So I want to have some clarification. So at the beginning, you said there's a parent line cross to make these lines, right? To understand all these phenotypes, try to connect with genotypes. So how do you choose these parental lines to begin with to make cross?
It depends what your goal is. So for the breeding purpose, we're trying to develop breeding populations. We want to maximize.
Because it's yield, right?
Right.
Right. So yield is the most important thing. What are the traits that you stick with?
Yeah. So the first thing related to yield, we want to develop populations that have a high mean, but they also have a high genetic variance, meaning that you have enough genetics to make your selections for it. If you only have high mean, okay, you have high yielding progenies, but you're not making any gain over your parents. So they need to combine both of those.
So both parent, high yield, but
genetically diverse, correct. And then second layer of decisions go into specific traits. So now with molecular markers, I know exactly if my parent is, you know, nematode resistant or stem canker resistant. So if I have, for instance, high yielding and high diverse potential parents, but both of them have, you know, let's say big A for nematode resistant, I'm going to have that specific allele fixed in my progeny. So that's one of the things we take into consideration. Now, for mapping purpose, you go in different directions. You want to cross segregating parents for that specific trait. You observe the variation on the progeny, and that's what you use to make your mapping decisions. Yeah.
I have a couple other questions. Can I continue or any other people?
I think so.
So you said machine learning, you have two types, right?
Mhm.
One was more accurate. Why is that?
Hard to tell.
Okay.
I think it depends a lot on the genetic architecture of your trait. We've seen that with random forest and molecular data, it can kind of handle a little bit more the dimension of the markers compared to something like a support vector machine. But you may use a different data set and get different results. I think the difference between the models are small. And at the end of the day, you know, if you have 0.79 or 0.77 accuracy, I don't think it's something to be too alarmed about. To me, the main important thing, number one, is you have good phenotypic data, so your model is accurate. And the other one is what's your application of the model. I don't know if the specific algorithm makes that much difference. We haven't seen that.
Last question, sorry. So GWAS data, you show that there are three peaks in Manhattan plot. What is this chromosome 15 one that you didn't understand?
We believe it's a false positive association. So that's a problem you run with GWAS. You get some peaks that don't necessarily make too much sense, especially if you're doing like single locus analysis. So it's almost like a t-test, right? You have one marker. Is it 1 or 0? How does that affect the phenotype?
You find any like ones that seems positive or true, but cannot explain from this known function, predictive functions.
No. I didn't have it here. So when we run the breeding lines for the GWAS, the one in chromosome 19 is a very conserved region. We have dozens of glycosyltransferase genes, so it's not just one. The one in 10 is very rare in diverse succession, so it's lost in modern soybean. That's one of the things we're trying to do, is bring that genetic source into modern varieties. And in the breeding lines, we found another one that makes sense in chromosome 6. So ideally, we would have a combination of all those regions and see how much you can explain from the trait. But it's in the making. That's what we're doing now. Yes.
So I get that all of your exposure was just kind of off target, but did you try to control that, or was that just incidental?
Yeah.
So thinking about like the timing of exposure and if that was.
So we refer to as chronic exposure during the season. We got exposed from day one until, you know, early reproductive. Very consistent, especially in that part of the state. What we try to do is, in the field, it's almost impossible to control it. We have greenhouse experiments with the extreme phenotypes, and then we play with different dosages, like 1/100th, 1/1000th, and so on. And it can kind of reproduce what you're seeing, not as well, because I think a lot of this is the ability to recover once the stress is you know, removed from the plants.
So what are the current restrictions in this area for the timing of that?
I think it's
followed, or?
I don't think so. No. There's the main problem is, which doesn't make much sense, is the cutoff dates for soybean are different than cotton. So even if you stop spraying for soybean, you're going to still be spraying for cotton. So they don't really work, the cutoff dates, no. Yeah.
So when you were answering Tomo's question, you said you thought the hit on chromosome 15 was a false positive. Why do you think that?
We look into the genomic region, it seems very empty. There's nothing we think linkages disequilibrium, both upwards or downwards. We look at the phenotypic variation, it didn't really seem to make any difference. We're really not sure why it was picked. It could be something related to the environmental interactions that we added, but it really doesn't make much sense.
One thing I would wonder is if, because when you look at those genomic regions, it's based on a reference genome, which came from basically the sequence of one accession. If you went in and looked at the did resequencing of the genome for resistant
Mhm.
infections, what if you saw genes in there?
It's possible. But then I would expect that the phenotypic difference would be more severe between, susceptible and tolerant based on that chromosome 15. It could be more on there. I mean, I'm not saying there isn't, but
What's the phenotypic difference?
almost non-significant. That's why we're not sure even why it was picked in the hit. It could be something to do with the allelic frequency between them, but if you look at the phenotype of the two groups, and then goes what we talked yesterday. It could be something that when you're interacting with something else and you're combining, several loci, it can show up and be, make a difference. But we haven't went that far. No.
Thank you.
Thank you. Yes.
So the phase 2 metabolism is a pretty generic detoxification mechanism. I wonder if that line would have, resistance to other herbicides that there are issues with, or
Yeah.
is it primarily dicamba that?
That's one of the things we were discussing. Because of what you just said, we want to spray them with different herbicides and see how they perform. We haven't done that yet.
Yeah, like 2,4-D might be one, 'cause it has a carboxyl.
Right. We want to see, and the one reason we didn't do it is because if you spray dicamba label rate, they're all gonna die. I mean, in our population, the purpose is not to make them resistant to dicamba. The purpose is to make them coexist when they get this minimal dicamba exposure. So I wasn't really sure how needed this would be for a 2,4-D, for instance, for practical applications, because you don't get that much volatilization. But for research purpose, for sure, yeah, it's on the makes.
It would just be interesting.
Yeah.
And then also I was wondering if there were any cytochrome P450s that were significant, or was it just?
We didn't find that. And one of the things that I would imagine is that is somewhat fixed in soybean. Because when you look at them, most of the distribution, they lay within this moderate. So the very susceptible and very tolerant are kind of extreme phenotypes. So it may be the fact that they're somewhat adapted already for that.
Well, the other thing is that dicamba already has a carboxyl group on it, and then the glucuronyl transferase can directly conjugate that. So you may not need it.
Right.
Just check if we have some questions in the
No.
Any one last question?
So with one last thought.
Yeah.
Is so a lot of times people in weed science, when they want to know if herbicide resistance is metabolism based, they'll use something called, it's an insecticide called malathion, and it knocks out cytochrome P450 metabolism. So you could just spray that on your plants,
That's good to know.
expose them to dicamba and see if they're still resistant. If they lose that resistance, you really know it's probably metabolism.
I didn't know that. That's good. Okay. Yeah, one of the things we start working this year is looking at the dicamba metabolites and see what's happening between tolerant and susceptible. It's very preliminary data. I mean, there's not really much to share, but it seems like you can see differences in the response, especially after spraying between tolerant and susceptible for a few key markers that we're using. But this thing about the insecticide, I didn't know that. That's nice.